talk. Unfortunately, the speaker could not make it here, but we have Ying from University of North Carolina.
Thanks for introduction, and I'm really happy to be here to present this project on behalf of Ying Huang and UNC Brain Lab. So let's start with some basic background on this project. So during the neonatal period, the cerebral cortex undergoes really rapid and critical development, which is pivotal for future behavioral and cognitive abilities. And some previous study has shown that cortical surface area expands significantly during the first 2 years of life, with most changes happen during the first year. And the same trend has been observed for cortical thickness as well. And in this rightmost chart, as we can see all the curves, so most of the significant changes in cortical features happen actually prenatally and immediately after birth, sometimes within the black rectangle. And genetically factors are major determinants in shaping neonatal cerebral cortex. As shown on the left figure, genetic influences on cortical thickness, CT, and surface area, SA, are actually different, and the influence on each brain area also varies. And in our previous work published last year, we successfully identified distinct genetically influences on the spatial patterns of cortical properties in neonates. So we got this genetically informed parcellation maps for cortical thickness and surface area. However, we realized that our previous work have predominantly focused on a coarse scale. So each region is pretty large. So, and each map only focused on one of the cortical property. And so we realized that these cannot adequately represent the fine-grained and multifaceted nature of genetic influences on cortical development. So we started this project to obtain a more detailed and comprehensive depiction of how genetic factors regulate early cortical development. So in this project, we aim to generate a joint, fine-grained, and genetically informed parcellation map of the neonatal cerebral cortex. And we are using the exact same data set with 202 neonatal structural MRI scans with both dizygotic and monozygotic twins with same sex, and their demographic information is shown here. Ages are in terms of postmenstrual weeks. So first of all, we need to process all of our images using the IB, the version 2.0 pipeline. Is available at the URL below all of the images. So I'm going to go through the major processing steps right here. So first of all, we need to correct any inhomogeneity caused by intensity difference. And then we did skull stripping, removal of cerebellum and the brainstem. And after that, we did tissue segmentation into white and gray matter and CSF, and filling the non-cortical structures and separate the left and right hemisphere. We also did topological correction and reconstruct the inner and outer surface with these triangular mesh. And we also calculate the middle surface as a geometry average of the outer and inner surfaces. And because we are interested in cortical thickness and surface area, we then went on to calculate these cortical attributes. Cortical thickness was computed as the minimal distance between the inner and outer surfaces, and surface area is the average area of all of the triangles associated with a single vertex at the middle surface. And because later we are going to do a correlation calculation, so it's really important to establish vertex-to-vertex correspondence between different scans. So we mapped all of the inner surface sphere onto a UNC 4D neonatal and infant cortical surface atlases. The URL is also below here and is publicly available. And And then we begin our calculation after obtaining the smooth and downsampled cortical thickness and surface area map of all of our skins. And we arranged all of the skins for both left and right hemisphere together to obtain the feature matrix. And here we include only 4,628 vertices, excluding all the vertices in the middle wall because those are not very significant for surface attributes. So then we separately calculate the genetic correlation matrices for cortical thickness and surface area between each pair of cortical vertices using the standard bivariate ACE twin model. So the breakdown of ACE is shown up there in the green box, and this model can help us to estimate how much variation in a phenotype. So here, cortical thickness or surface area is accounted by genetic effects. So the bivariate version of the ACE model can help us estimate a pattern of genetic and environmental variances and covariance. And after getting variance and covariance, we can calculate correlation matrices and for both of the cortical attributes. And then we put those matrices into multivariate spectral clustering analysis and to group all vertices into k distinct different regions, and each region are consisting of a group of vertices that are maximally genetically correlated. So how do we determine which is the optimal region number? So here we adopt 3 different measures. So first of all, the Silva coefficient for cortical thickness and surface area, and we also adopt 2 of the measures for measuring clustering efficacy, ARI and normalized mutual information. And for all of these measures, the higher the value, the better the correlation results. So we are looking at where the local maxima for each overlap. And so we can see from the plot, this actually happened at k equals 35, 67, and 86. And then we went on to visualize all 3 candidate results. And after considering the spatial distribution and hemisphere symmetry of parcellation maps, we settled down to use 86 regions. And finally, to enhance anatomical consistency and comparability of corresponding regions in both hemispheres, we combined bilateral symmetric regions and assigned a shared ID for each of the region. So we are getting this final fine-grained and genetically informed parcellation map of the neonatal cerebral cortex. And the numbers shown here are the ID that we assigned to each region, and the names shown below is the approximate name for each of the region. And we are happy to find out that all of the regions exhibit not only bilateral symmetric patterns, but also correspond really well to structurally and functionally meaningful areas. So to sum up, our contribution is to reveal this fine-grained and genetically informed parcellation map of neonatal cerebral cortex combining multiple cortical properties. And also this parcellation map can comprehensively reflect genetically regulated detailed patterns of neonatal cerebral cortex and are structurally and functionally meaningful. And these maps can be found on our website as well. So that's all I have. And if you have further questions, please feel free to contact Dr. Lee. Thank you so much for your attention.
Thank you.
Thank you for the talk. I think you built the parcellation with a lot of cortical features, and I can agree that it might be structurally relevant, but how did you demonstrate it's functionally relevant?
Because I'm not part of the author for this project, so I'm not really sure, but you can always contact Dr. Lee and ask them. I'm sorry.
Are you willing to answer more questions?
I can try my best to answer as much as I can, but yeah.
Any questions from the floor? If not, I'm on the floor. Just have a quick question about, because in an infant stage, we often see that the contrast flipped, right? If you go to the one slide showing this pipeline. And yes. I was wondering how your pipeline deal with this contrast flipped for segmentation. Like from the skull strip, the first two, right? You can clearly see that the white matter is, well, not close to the CF, close to the cortical regions. The white matter is supposed to be lighter, and gray matter is supposed to be darker in the T1 image, but currently it's flipped. This is the big issue for the early developmental structure image.
To my knowledge, in the pipeline, we first aligned T1 and T2 weighted image before doing skull stripping, so I don't know if that will help, but that might be answering your question.
Thank you.
Thank you so much.
Thank you everyone to join this session and enjoying your last day of OHBM.
And please remember to fill out the evaluations online.